today's seminar, Nick Schulte, who's a postdoctoral fellow at the University of Colorado, will be speaking on application of diatom multimetric indices to state monitoring data. I'm really pleased to introduce Nick. Nick has an incredible and high level of skill in a large number of areas, including he's developed algal-based DNA barcode methods. He's developed DNA bioinformatic pipelines for consistent analysis of DNA data, both under taxonomic and taxonomy-free approaches. Nick uses modeling to understand natural and human influences on both cyanobacteria and diatom distributions. And he's currently working on testing the efficacy of predictive O/E modeling, which is observed or over expected modeling in paleolimnology. And Nick is looking for a job right now. And so if you don't snag him right now, he's going to be gone. Act fast. His postdoc will be up soon. So today Nick will be presenting in a little bit of a different format in that the talk has been recorded. So Nick can ask himself his own questions as we go. So be sure to make them good ones, Nick. So I would like to introduce Nick Schulte.
Excellent. Thank you, Sarah, and welcome everybody. I'm excited to be here. As Sarah said, I'm going to be playing a video recording, which is just my voice, walking through a PowerPoint presentation as well as some video tutorials. So I will keep my video on, but I won't be speaking, and I don't have it memorized, so I won't be lip syncing either. So I hope it's not too distracting. If so, then say so in the chat and I'll turn my video off. And I hope you all enjoy the presentation, and I'll of course be around for questions afterwards. Thanks. Hi everybody, my name is Nick Schulte and I use he, him pronouns. I'm a postdoctoral researcher at the University of Colorado Boulder, which is located on the traditional territories of the Arapaho, Cheyenne, Ute, and other indigenous nations. First, I want to take a moment to acknowledge the painful history of genocide and forced removal from this territory, and emphasize that I honor and respect the many diverse indigenous peoples still connected to the land on which this work was performed. I'm excited to share with you today some work that was done with my colleagues Sarah Spalding and Sylvia Lee on applying diatom multimetric indices to state biomonitoring data. And for this presentation, I'm going to use the example of work that was done in and with the state of Colorado. In doing so, I'll walk through the entire process of using a publicly available web-based tool that is applicable to streams across the conterminous United States. I'll provide some tips and tricks for you to use your own tribal, state, regional, or national monitoring data in order to compute scores of disturbance to diatom biotic integrity in streams. This work was done in collaboration with the University of Colorado Boulder, the Environmental Protection Agency, and the State of Colorado Department of Public Health and Environment. So a quick outline, I'll start by just defining what diatom multimetric indices, or MMIs, are, then walk through the development of some regional MMIs that are broadly applicable throughout the United States and resolve issues of previous indices, which is why we're going to be using these MMIs in this example. Then I'll walk you through how state diatom data from the state of Colorado were applied to these regional MMIs, and finally talk about how we evaluated the resultant MMI scores relative to state water quality measurements. So why the focus on diatom multimetric indices, or MMIs, in the first place? Well, MMIs are a commonly used tool for aquatic bioassessment, yet there's been a limited adoption of diatom-based MMIs or any other tool across the US. This is in large part because of three major issues. First, taxonomic inconsistency across labs and programs obscures important patterns and reduces the performance of many existing tools. Second, tools like MMIs use traits or ecological attributes of diatoms and many of those attributes are based on European taxa, which doesn't translate all that well to the North American taxa that we're concerned with for state and national bioassessments. And third, there's often limited applicability and/or accessibility of existing tools. The existing tools, like those MMIs that are already out there, have in many cases not been tested for state use and/or are not controlled enough for natural variability within states or regions. Also, they often require a lot of technical expertise to develop, test, use, which is critical, and interpret. So multi-metric indices, or MMIs, have been one of the most widely used tools for bioassessment in general, and for diatoms, they tend to be the most used tool where bioassessment has been attempted for states or at a national scale in the US. So what are MMIs? MMIs are basically the sum of standardized metrics, and the metrics are ecological attributes that represent unique features of the diatom community and are also responsive to anthropogenic disturbance. And we can think of metrics as being part of different categories, including diversity, such as diatom taxon richness or relative abundance of certain taxa or taxon groups, traits such as their diatom habit or the motility of diatom taxa, environmental preferences such as how well they tolerate pollution, nutrient concentrations, or salinity, and fourth, taxonomy, which general taxonomic or phylogenetic group are the diatoms in. And in general, we can interpret the final MMI scores, the sum of those metrics, as low scores indicating harmful effects of disturbance, of anthropogenic disturbance on the biotic integrity of diatoms. So several diatom MMIs have been developed at different scales, from within states to entire states to across the entire US, but they have limited availability across those different scales and a variety of different checks and developmental procedures. And as such, there are a lot of unresolved methodological issues in the available diatom MMIs, mainly the number of component metrics that are used in the final MMI, whether the MMI accounts for natural variation across the spatial extent used, how well the metrics responsiveness to stressors is known, and then how the final metrics of the MMI are selected, and then finally how accessible that final MMI is to water managers, environmental managers who need to apply and generate these MMI scores to environmental assessment. So this is where the not just another diatom MMI comes in. Darren Carlisle with the U.S. Geological Survey, along with colleagues in the EPA, University of Colorado Boulder, developed 3 regional scale diatom MMIs that cover the entire contiguous US. So these MMIs were generated from 1,500 sites collated from the USGS and EPA national surveys. As I said, they spanned 3 separate regions: the Eastern Highlands, the Plains and Lowlands, and the West. And then there were 3 independent data sets from the USGS that were used to assess the metric responses across these 1,500 sites to specific stressors, water chemistry stressors, watershed regional stressors, hydrologic stressors, things like that. Carlisle et al. Considered 61 different metrics across 13 categories when generating these MMIs. And one key component is that within each of these metrics, natural variation was modeled out if it was determined to have a strong influence on the metric score distribution of the sites within each region. And thirdly, metrics that were sensitive to taxonomic inconsistency were removed. And we can see here on the left that along the x-axis, we see whether basically a measure of taxonomic inconsistency, how much the diatom analyst influenced the distribution of or explained the variation in metric scores for these different metrics. So for instance, organic enrichment, the metric organic enrichment was heavily influenced by this analyst effect, or heavily explained by this analyst effect, whereas general habitat at the bottom of the y-axis had almost no analyst effect. So in this MMI, Carlisle et al. Chose metrics that were on the low end, the bottom part of this y-axis, that were not heavily influenced by analyst effect and taxonomic inconsistency. Furthermore, Carlisle et al. Looked at how the number of component metrics in the final MMI influenced the sensitivity or the performance of that MMI. And if we look on the here on the right, for each of the regions, the Eastern Highlands, the Plains and Lowlands, and the West, there's more or less an asymptote that happens at 6 to 7 component metrics in the MMI. So the highest MMI performance or ability to differentiate reference in disturbed sites was optimal for more component metrics, between 5 and 7 component metrics within each region. Then, to ensure that the metrics that were selected actually represented ecological responsiveness of diatoms, Carlisle et al. Looked at how sensitive the metrics were to stressors using independent data sets. So for within the West region, for instance, an independent data set in the Pacific Northwest, which had water chemistry data, stressor data, etc., was used to see which stressors most explained or were most associated with the metric that was in the final MMI. So here's a small example for a metric. I'll define this in a moment, but for now, this BC4 metric had some natural predictors, and that variation was modeled out. But then in this independent data set, they found that total phosphorus sediment metal concentration and time since peak flow have strongly influenced or was associated with the distribution of this BC4 metric score, to the point of 0.46 or 46% of the overall variation being explained by these 3 stressors, suggesting that BC4 is responsive to certain types of stressors. Therefore, when it's incorporated into the MMI, that MMI is also integrating that aspect of the community responsiveness to those stressors. And finally, what sets these MMIs developed by Carlisle et al. Apart from previously constructed MMIs for the US is its public availability via an R-Shiny web application, which is accessible via these 2 links put here. And basically, this provides a web interface where state water managers or other interested parties could upload their own site and diatom count data and have the web application compute MMI scores for you and allow you to take those MMI scores and analyze them by themselves, the distribution of those MMI scores, or alongside other data you might have available, such as water chemistry data and the like. Here is a brief overview of the interface of the Diatom Assessment Tool MMI Shiny application website. So the first page that you see is the Introduction tab, where it just walks you through what this tool does, what are the basic requirements, and then how do you use this tool. The final tab on the top right are detailed instructions that walk you through all of the specifics that each step in the process requires. Your data, what your data look like, how the MMI is calculated, and then how you can interpret those MMI scores. And then to actually upload your data, you go to step 1, upload site information, where you're going to upload your site list, which we'll talk about in a moment. Then upload your taxonomic data once you've uploaded your site information. Then calculate your final MMI scores. And then finally, there will be a box plot that will be presented after the calculation of your MMI scores to help you in data interpretation. And we'll be going through all of these steps in more detail, but this is just a general introduction to what the web interface looks like. So for the rest of this presentation, I want to provide both an example of results from applying state of Colorado count data to this regional national MMI tool, as well as provide you tips and tricks for performing this by yourself. So briefly, for the state of Colorado, benthic algae have been collected since 2004, over which time 406 diatom samples have been counted, 351 of which have water quality data associated with them. So we applied these 406 samples to the MMI tool to compute MMI scores, and then related those scores to this water quality data. The first step in the computation of MMI scores using the Shiny app is to assign what are known as COM IDs to each site that is associated with a sample. COM IDs are unique identifiers of stream segments in the National Hydrography Dataset, and they're used in the MMI computation to model out or account for natural variation among your sites. Therefore, it's critical to have accurate COM IDs for each of your samples. You can get COM IDs from your site latitude, longitude coordinates by manually searching for each site by following the link shown here, or by using spatial join features in ArcGIS or lookup functions like those in the NHDR package in the R software environment. I won't go into more detail here, but you can follow a tutorial video I created on how to do the manual lookup, which will be posted on the diatoms.org project webpage. For the state of Colorado analysis, we manually assigned COM IDs to each site, which ended up being one of the most time-consuming steps in computing MMI scores using the Shiny app. But regardless of whether you use manual or automated COM ID assignment, it's important to verify that your COM IDs actually match your sample site. Therefore, there's a QA/QC step in the Shiny app to input user-calculated drainage area of each site and compare it to the drainage area calculated from the National Hydrography Dataset based on the COM IDs you just assigned. The Shiny app will then flag any sites with a large difference, 30% or more, between the two drainage areas as an indicator of potentially misassigned COM IDs. If you're confident in the assignment of your COM IDs, this step isn't strictly necessary, and you can give all of your sites a drainage area of some other arbitrary number and ignore the results of the comparison. However, given human error for the manual method and limited oversight for the automated methods, I recommend using this step to double-check your COM IDs. Otherwise, the MMI scores may be inaccurate because of improperly modeled natural variation. Now, many state and regional programs already have drainage area calculated, in which case this step is straightforward. For those without readily available drainage areas, I provided a tutorial video I created on how to calculate drainage areas on a site-by-site basis, as was done for the state of Colorado in this analysis, which will be posted on diatoms.org, just like that video for the COM ID assignment. So altogether, once you have your COM IDs and your self-generated drainage areas for each site, you'll come away with a file that you'll input into the MMI Shiny application, in which columns are sample ID that in which each sample ID has a corresponding COM ID and drainage area. To upload your site information into the Diatom assessment tool, you need a site information file that contains COM ID in one column, a unique sample ID in another column, and then your upstream drainage area in square kilometers in another column. They all have to be labeled specifically COM ID, sample ID, and area. So in this example, I'm looking at 10 different sites based on these sample IDs. And I've gone ahead and created a couple of errors that we'll be able to see in the Diatom Web Assessment tool. First being that in this first sample, 1, there is no COM ID, or the COM ID is 0, so we'll see if that throws an error. And in the second one, I've changed the area from 593.6 to 5,593.6 as an example of a typo in the area, even if the COM ID is correct in this situation. And then these all should correspond, these COM IDs all should correspond to the sample that we're interested in, meaning that the areas that we've calculated should be somewhat similar to the areas calculated by the NHD database. So to upload this information into the Diatom assessment tool, you go to the web app and you go to the tab Step 1, Upload Site Information. You then go to Browse and you find the folder that you're looking for your site information. And the biggest thing is to, in this drop-down, at least in Windows computers, to go from custom files to all files. And here I'm going to select this demo MMI site information CSV file and click open. And you can see the upload is complete, and on the bottom right that it's the status of it extracting the NHD watershed features, which will be used to model out natural variation in your sites. And here a warning comes up that says there may be COM IDs that are not matched to NHD COM IDs, and the number of sites where this occurs is 1. We can see what those sites are by toggling this box called List Sites with Missing Predictors due to COM ID issues. And indeed, we see that sample ID 1, which has that 0 COM ID, is throwing that error. We can also see areas with basin or samples with basin area QA issues, meaning that the basin area doesn't seem to match the NHD area. So for the first one, this is the one that I artificially increased the area of from 593 to 5,593. You can see here that it says, wait, you might want to double check that this is the correct COM ID because your area is so different from the NHD drain square kilometers. Of course, I know that this is merely a typo, so that can be fixed by just removing that 5, but it would be worth to go back to the NHD web map and looking up this COM ID to make sure that we did indeed select the correct area, and maybe double checking our calculation of that area. Interestingly, another COM ID threw an area QA issue, in which case this area is, quote unquote correct. We calculate it as 112.1, but the NHD calculates it as 242, so nearly more than double what we calculated. This just means that we should go back and make sure we got the correct COM ID, but maybe this is just a difference in how areas are calculated between the two methods as well. So you might choose to ignore these flags, but they are there for your own information as to whether you want to proceed with the COM ID you selected or alter it to make sure that it's consistent with your samples. And again, the ones that are flagged here under the basin area QA issues are sites with greater than 30% difference between the NHD drainage area and the user supplied drainage area. We can choose to move forward as is. The areas that we calculated are not going to be used further. It's purely for a QA check. It's only going to use this drain SQKM variable going forward. So we can move forward with this if we don't care about double checking our COM IDs, and we can move forward if we don't mind this sample being dropped from further analysis. And we can see all this by downloading the complete predictor data, in which case we see our COM ID, our sample ID in our area, what we input, and then everything that's calculated from the NHD that will be used to model out natural variation and metric calculation. We can see here that that one sample ID, number 1, is dropped from further analysis. So as long as you're okay with that, then we can move forward, but you might want to go in and get the correct COM ID for that sample 1 so that you can actually get data back for that sample. And that is the process of uploading your site information into the Diatom web assessment tool. Following the preparation of your site information, the next step is to prepare the Diatom data. The biggest thing here is to make sure your Diatom data are in the form of raw counts or relative abundance, and that the data can be, the taxonomy that you use for your Diatom counts is comparable or can be harmonized to the list of Diatom taxa that's used in the MMI. So basically that list is all the taxa and all those names that have at least one metric value across the 3 regions. And that state taxonomy is largely based on USGS biodata name, so should be updated. So the goal here is to update your Diatom taxonomy to the MMI Diatom taxonomy so that the two can speak together to each other. So what this step does is it allows you to have the opportunity to update the taxonomy the state is using to that of which the MMI uses by giving you a series of flags and checks that you can re-upload revised data. And after you have updated the taxonomy as best you can, it will then remove observations of unmatched taxa from the state database from which the MMI will be computed. So the input file that's required for this step is long format diatom counts associated with unique sample IDs that are the same as those in the site information file. The second step of using the Diatom web assessment tool is to upload your taxonomic data. These data should be in long format with your unique sample ID in one column, your taxon names in another column, in which your taxon names are separated by underscores between genus, species, variety, and the like, and then your counts in the third column. These can be either raw counts or relative abundance counts. Then you go to the Diatom web assessment tool and go to step 2, upload taxonomic data, and click browse to upload your file, which should be in CSV form. Just as with the site information, you click custom files or you click the drop down to select all files instead of custom files, and then select your CSV file that corresponds to your taxonomic data, which I've named as such, and click open. Once your upload is complete, you can then see a list of all taxa that are unmatched between your taxonomic data and the taxon names found in the metric database that's used in the MMI. Basically, you want to have as many of your taxon names correspond to as many taxon names with metric values in the database, in the MMI database. So you can see a list of unmatched taxa by clicking the box. And we see that in our sample data set, there are 3 taxa that don't have names that corresponding to names in the MMI database. And these are Acanthes minutissima, Adelphia minuscula, and Cyanema temperae. So with a little bit of diatom knowledge or just a little web browsing, you can identify some potential issues with these. For this Acanthes minutissima, you could go to websites like diatoms.org or algaebase.org, type in this name, and you'll find that it's an old synonym of the currently accepted name of Acanthidium minutissimum. This Adelphia minuscula, if you type that in, it will likely autocorrect to Adlaphia minuscula, in which case we basically have a typo here between the A and the L that we need to correct for one of our samples. And then this Cyanema temperae is a currently accepted name of a taxon, but it does have some older synonyms that could be represented in the metric database rather than this updated name. I'll note that that's unlikely to occur because the metric database usually uses the most updated names, which corresponds to the USGS dataset, and so using older synonyms is unlikely to be helpful, but it might be worth a shot if you're losing a lot of observations across a lot of samples. We can see how many counts and observations we're losing if we dropped these taxa by looking at a list of samples and counts after taxonomic harmonization. So if we click that, we can see that across the 10 samples, some samples aren't affected by these name discrepancies at all. Basically, sample 1 does not have any 3 of these taxa, therefore none of its counts are being dropped, or none of the taxa are being dropped from the MMI calculation. Meanwhile, sample 10 has about 5% of the counts being dropped, and sample 4 has something about 13% of samples being dropped, or of taxa and counts being dropped. Sorry, of counts being dropped, not of taxa, not of samples. So if we want to fix this, we can go back to our database and look for any observations of Acanthes minutissima and replace it with the synonym Acanthidium minutissimum that is likely to be in the MMI database and change all those. And then we'll look for that Adelphia, and we can see here there are other observations of Adlaphia minuscula, and there's one typo here in sample 4, so we can just correct that typo. And as for that Cyanema temperae, we can now see if it's going to substantially affect the counts or if we're okay with that one being dropped before we do all those synonym checks, which you're definitely welcome to do. And then you might want to save this as an updated file rather than overwriting your old file by adding an updated qualifier to the name. Then you can go back to your Diatom assessment tool webpage, upload the new updated file, again, clicking all files first. And you see that now it's updated so that Encyonema temperae is the only taxon that doesn't match, and that it's only found in one sample with 1% abundance. So if we're okay thinking that 1% abundance in one sample is likely not going to substantially affect that sample's MMI score, then we can proceed without testing different synonyms for temperae, because it's possible that this is a more regional taxon that doesn't have a metric value nationally, for instance. So we can then download the final harmonized taxa file that will be used. It doesn't re-relativize sample counts. It just provides you what's actually being used by the MMI. So it's very similar to what we've provided it, except you can see if you search and find Encyonema temperae is no longer in this file. And that is the process of uploading your taxonomic information, adjusting it based on synonyms to harmonize it with the MMI names, and then the final step is to proceed to step 3 MMI calculations with your harmonized taxon database. So for the state of Colorado, at this step, there were 74 unmatched names originally, and following harmonization done by Sarah Spalding and myself, 23 taxa were determined to not have any synonyms within the MMI database. So those 23 taxa and the counts corresponding to them were removed from further analysis. And across all of these 23 unmatched taxa, they were all present in less than 6% of samples each, and 96% of samples had fewer than 10% of counts removed. So we feel fairly comfortable that not a substantial number of counts were removed by losing those 23 unmatched taxa, that our results are still valid despite not having those 23 taxa reflected in the MMI scores. And then finally, once you upload both the site information and the taxonomic data, the shiny app will do the rest of the work for you. It will compute the MMI scores by modeling out natural variation based on the COM ID. It will also calculate values for each of the 61 metrics, and then sum the metrics scores from the component metrics in the final MMI to give you those final MMI scores. So what is the composition of those regional MMIs? And for the state of Colorado, sites were found both in the west and in the plains and lowlands region. So that's why I'm showing you the metrics that comprise each of those MMIs. And I won't go into too much detail here, but generally in the west there was a metric that called BC4R-Res, which represented biological condition and tended to be in more disturbed sites. So a higher score of BC4 indicated an increasing response to disturbance. And what the R here represents is a richness-based calculation, and the res represents that natural variation was modeled out. Additionally, there was a metric for high nitrogen taxa, high salinity taxa, weakly modal taxa, taxa present in the Acanthididae group, and then stalk taxa. So a variety of categories represented, and each category had, represents either increasing or decreasing responses to disturbance. On the plains and lowlands, the biological condition metric was one indicating low disturbance relative to the high disturbance indicator of the west, as well as low phosphorus, high salinity, highly motile, and centric taxa. So we're now ready for step 3, actually calculating the MMI scores for each sample that we input. We can go back before doing anything and just double check that what site information we have available. So this is what we've already done, where we have one site without a COM ID, 2 sites that have some potential QA issues, but we've decided to ignore those. We also haven't changed that site with the COM ID issue, so it's still there. We've adjusted the taxonomic data and harmonized it to the MMI database taxonomy, and find that we would only lose 1% of one count, or of one sample. So we can double check to our data to see this information here in the step 3 MMI calculations tab. We can click data check, and it says that we're missing biological data from 0 sites, and that one of the biological samples has missing environmental data, and that's that COM-ID one. We can see the list of sites with missing biological data is 0, and the list of biological samples with missing site data is 1. Next, we can compute the metrics. So we can compute the metrics, and these are all the metrics that are in the MMI database. They're not necessarily used for the MMI, but they're all the metrics that were used in the construction of the MMI, which you can use for your own purposes down the road outside of the MMI results themselves. You can click Download All Metrics to see for each sample what the score is for each metric that we have available as part of this MMI endeavor. So here for the BC1 richness metric, you can see that there's a score of 3 for sample 1 and a score of 8 for sample 7. And I'm not going to go into interpreting any of these, but you can look at the manuscript that this paper, that this tool is part of to better understand what those metrics mean. You can then, the next step is to actually compute the MMI scores following the computation of the metrics. So you click Compute MMI, and it will have a loading bar here on the bottom right, which went quickly here, we might not have seen it. And then it tells you that the computations for the MMI are complete, and then notes the number of sites in each of the 3 regions that there are separate MMIs for. And in this case, all 9 of our sites, because that 10th one without the COM ID was dropped, all 9 of the sites that were used in analysis are found in the western region. Therefore, the West MMI was used for those sites. So in that case, we can download the output of that West MMI here, open it up just to look or to analyze in your own way, and you can see the calculation of each of the metrics that are in the MMI. You can identify the 6 metrics that are present in this MMI, and then the final MMI score for each sample here. So you can see that site 10 has a total MMI score of about 30, whereas the site 8 has an MMI score of 54. And generally, these MMI scores go from low to high, being from most disturbed to least disturbed biotic integrity of the diatom community. And we can look really briefly to see what distribution of samples, sample locations would be within each of those 3 MMIs. And now we can go to the MMI interpretation tab to get some sense of the distribution of those MMI scores. So we don't have samples in the plains and lowlands or East Highlands, so we go to the West tab. And here there's a little note that says that generally sites with MMI values less than the 25th percentile, which is noted as the horizontal line here at the bottom of this box plot, may be biologically impaired, or if that terminology isn't suitable for you, more biotic disturbance or biotic integrity may be disturbed. And a stricter rule of thumb may be the minimum here. Below the minimum might be the most likely to be disturbed. And what this box plot represents, apologies for not getting to that first, is the distribution of reference sites that were used in the creation of the MMI. So the box plot is the distribution of the national reference sites in the western region when the MMI was created. So this score of about 40 is the lowest MMI score that a reference site had when the MMI was created, whereas a 90 was the highest, and the average was about a 60. We can see that all of our sites are below the 25th percentile, which I suggest is potentially or possibly disturbed. Diatom biotic integrity is possibly disturbed, and anything lower than the minimum is likely disturbed. And there could still be work done to interpret those distributions more fully, but that's the general rule of thumb that's used in this MMI tool itself. And that is the final computation of the MMI. And again, you have this Excel spreadsheet where you can use these MMI scores alongside your sample ID and any other information you have associated with those sample IDs, like water chemistry, to further analyze and interpret what those MMI scores mean for your sample data. So the output for the MMI scores for the state of Colorado were that 367 samples were found in the region characterized as the West in the MMI, and 39 samples in the plains and lowlands. So once the MMI scores are computed, we want to be able to interpret those relative to some level of disturbance of diatom biotic integrity. And this can be done by comparing our observed MMI scores from the state to the MMI scores of reference sites used in the construction of the MMI. So here, these box plots represent the distribution of reference sites from the national samples that were used to construct the MMI. So in the West, the maximum reference site score was about 85, and the minimum was about 40, with an average of 65. In the plains and lowlands, everything was slightly higher based on the MMI. So that's what we use as our relative indicator of disturbed or undisturbed. We can break it down by saying that if we observe a point of our state data, let's say in this above the 25th percentile of reference sites, so let's say at 80, then it's likely to be undisturbed. If it's below the 25th percentile, but above the minimum of the reference site scores, it's possibly disturbed. And if it's below the minimum of the reference site scores, it's very likely to be disturbed. No reference sites were seen with an MMI score that low, which indicates once natural variation has been modeled out, that site score, that observed state site score, is more disturbed than any reference site in that region. So for the state of Colorado, we can look at each of our observed site scores, and they're just randomly distributed across the x-axis so we can see their full number. We see that 23% in the overall, between both the plains and lowlands in the West, 23% of all the sites were below the relative minimum reference site score, so we're likely disturbed. 39% were between the 25th percentile and the minimum, so we're possibly disturbed. And 38% of all the sites were likely undisturbed. Instead of just in box plot form, we could also plot these on a map using the same criteria of MMI scores being between certain percentiles, and see the geographic distribution of these possible disturbances. So here in the state of Colorado, we see a cluster of red-coded sites, or likely disturbed sites, in the northeast along Greeley and the South Platte River, which is a heavily agriculturally affected area. We also see an area here between Denver and Pueblo, slightly in the foothills of the mountains, whereas in the kind of the corridor of the high country mountains, we see some of these green and yellow sites. So we can qualitatively do a check whether those make sense geographically. And then the next step is to consider it relative to water chemistry. So in the state of Colorado, 351 of those 406 samples had water quality variables measured across all those sites. And we determined that there were 7 uncorrelated variables that we could attempt to relate to those MMI scores. So pH, conductivity, ammonia, total phosphorus, dissolved copper, dissolved iron, and zinc. The first step was to see whether the diatom taxa were distributed relative to these environmental gradients in expected ways. So we plotted it in the taxa and the water chemistry in an ordination space. And basically any taxon that's close to these arrows suggests that they are affected by increasing concentrations or amounts of that variable. So here, this Reucosphenia abbreviata, Nitzschia, and Amphibia are associated with higher total phosphorus in this state database, whereas this Navicula tripunctata, Rhopalodia gibba, are associated with higher conductivity. And these ones over on the far right are associated with lower concentrations of those variables. And in general, these relationships between taxa like Nitzschia, Amphibia with high total phosphorus matches or is consistent with previous research on diatom ecology. So we're just independent of the MMI doing kind of a gut confirmation that the taxon distributions are responding to environmental gradients in expected ways, which they did in the state of Colorado database. Next, we looked at whether the MMI, or whether the metric scores, not the MMI scores, are associated with these water quality variables in expected ways. So you'll recall that I I went through the table of those component metrics and noted which ones increased with disturbance and decreased with disturbance. So for instance, this high N metric should increase as nutrients increase. And as with the previous slide, it's closer to this increasing arrow of total phosphorus, suggesting that these high nitrogen metric scores are associated with high total phosphorus, which is expected. And similarly, here is BC1, which is a low disturbance. It doesn't like a lot of pollution. So these scores should increase as nutrients or stressors decrease. And indeed, we see it on the opposite side of this increasing total phosphorus gradient. So BC1 is not increasing with nutrients, which is the expected relationship here. Finally, we looked at how MMI scores themselves respond to specific environmental gradients. And this was done using generalized additive modeling, which allows us to see the additive effects of different water quality variables on the MMI scores, or the relationships between MMI scores and individual and additive environmental variables. So here, among the west sites, basically we interpret this by the x-axis being increasing amounts of that stressor, and then the y-axis being the relative change in the MMI score. So here we see that as total phosphorus increases, MMI scores decrease exponentially. And with conductivity, we see up to a certain point, about 2,000 microsiemens, that MMI scores decrease exponentially with potential increase at very high salinities. And that also dissolved copper seemed to have a positive effect on MMI scores. Among the plains and lowland samples, we see the similar trends with total phosphorus and conductivity, mainly that MMI scores are decreasing with increasing amounts of each of those stressors. The distribution of MMI scores across the state can be visualized relative to those main stressors in an ArcGIS web map. So here we see the map that I showed earlier of the general rating of disturbance and diatom biotic integrity based on MMI scores across the state. So we see green being likely undisturbed, yellow being possibly disturbed, red being likely disturbed, and we see that same distribution. Based on the generalized additive models, we know that total phosphorus and conductivity were the most influential or the most strongly associated variables with MMI scores across both the west region and the plains and lowlands region. And in both cases, MMI scores decreased as total phosphorus and conductivity increased. So we can just visualize this in a qualitative manner as a self-check or just an investigatory, exploratory analysis by plotting the total phosphorus values relative to the MMI score. So here I've loaded the total phosphorus concentrations and give them a size circle associated with greater magnitudes or greater concentrations. And we can just generally see that indeed more of these red dots, plains and lowlands being in the diamonds, west sites being in the circles, that the red and yellow dots tend to be more associated with the larger circles of phosphorus, kind of giving us that gut check of what the GAMs are showing us, while also allowing us to explore where specifically we see those patterns popping up. So in this case, in Colorado, we see a lot of high phosphorus in these northeastern regions along the South Platte River, where there's quite a bit of agricultural influence, as well as in south of Denver, and then scattered about as well. And we can do the same sort of thing with conductivity. So I toggle off total phosphorus and show the general sizes of conductivity. And again, we see kind of an overlap with total phosphorus, that we see higher conductivity in these more agriculturally or more urban influenced sites, as well as some both on the eastern side of the state and on the western side of the state. Yeah, we do see some interesting kind of negatives, if you will, of a high conductivity on this site, despite it having a high MMI score, showing that there are some site-specific patterns that may not be captured perfectly by the MMI score variable relationships. And again, we can overlay both of these together and see that there is some overlap in high conductivity sites and high phosphorus sites, but that they also are distinctly are showing distinct patterns as well, which is what the GAM allows us to do by looking at multiple stressors at the same time. One final thing we were able to do with this this data set is look at reference sites that have been determined by the state of Colorado and look at the MMI scores from an invertebrate MMI and see if that kind of maps to high MMI scores of diatoms. So if I toggle this reference site invertebrate MMI scores, we'll see that the larger circles indicate higher MMI scores. And these are only for pre-assigned reference sites in the state of Colorado. And we can see for the most part that a higher MMI score with invertebrates, again, these are all reference sites, corresponds to a higher green or yellow MMI score for diatoms, but that there are some potential discrepancies like this site here, which the diatom MMI score is very low on, yet it's distinguished as a reference site with a high MMI score for invertebrates. So this is the sort of analysis that could be done after the diatom MMI score by comparing to existing indices that states or regional programs have access to see whether the different biotic groups parallel each other in response, or whether there are different signals that we can pick up based on the different groups, which we see some agreement and some disagreement here on this very cursory qualitative look at the diatom MMI scores relative to reference site invertebrate MMI scores. So just a taster of further analyses that could be done depending on the data availability for different states. So altogether, this exercise demonstrated both really useful information for both the state of Colorado and any other state, regional, water managers, or ecologists who are interested in the biotic integrity of diatoms across a certain spatial extent in the conterminous US. So mainly, we found that the use of the SHINY application with independent state data for these new regional MMIs was straightforward. It's very straightforward to apply and to adjust state data to this national MMI web application. Within the state of Colorado, we found that based on these MMIs, that 62% of the samples across the state reflect possible disturbance of diatom biotic integrity, and that phosphorus and conductivity are the dominant stressors related to decreasing diatom biotic integrity. And with that, I would like to thank you all for your attention and invite any of your questions on the Carlisle et al. MMI, the SHINY application tool, or the Colorado results. Thanks.
Great job, Nick. Thank you very much.
Thank you, Mr. Fox.
I can see why you wanted to pre-record that for all the demonstrations. I can only imagine trying to pull that off. If anybody has any questions, feel free to raise your hand or type them in the chat. Nick's been doing a great job of answering them as he's gone along. I, of course, want to ask you, Nick, what would it take to adapt this tool to lake management and monitoring?
Yeah, thanks for that great question, David. And again, thank you all for your attention and apologies for the distracting tennis ball practice, but I had to get it in somehow. So I'm actually in that position right now, is looking at lake monitoring and have thought about applying an MMI approach to lake sediment samples. The biggest limitation in doing that is the difference in taxa and their ecological attributes / traits, so that the metrics values for even the same taxon in streams versus lakes might be different, therefore leading to a different interpretation of disturbance to diatom biotic integrity. So the biggest one is attributes differ between this MMI and likely lake MMIs, but also you have different environmental factors that are not picked up in the same metrics even.
So you might say then that maybe the template for the tool is good to go. You just have to do the legwork of generating new audioecological information specific to lakes.
Correct, and there would be I mean, there's a lot of process involved in building this tool of, you know, it's an all subsets approach. So there were with 6 to 7 combinations, up to 6 to 7 combinations of metrics. That was over 1 billion different MMIs that we had to choose from, and that required measuring different attributes of precision and sensitivity and performance, as well as just expert opinion for interpretability and applicability. So it's not just a plug and play process per se of, we have metric values for all these taxa, now we can just rerun this code and we're good to go. There is a little bit more legwork on the front end that would be needed, but it's something that I'm sure we would be, not to speak for Carlisle at all, but that code would likely be able to be made available in some capacity to those interested.
Cool, cool. And yes, as Sarah was saying, hello, Samaya. It's good to see you. Nick, I wanted to ask you one other question. Oh wait, here we have a question from someone else. Hera, I believe if I have your name correctly, asks, can we use this MMI tool for sites with diatom data counted for multiple years?
Oh, that's a great question, Hera. Please let me know if I mispronounce your name. This data set is from multiple years, and there are a couple of sites that are repeated over that time. So I believe it's from 2004 to 2017, we have those 406 samples from. But you're correct in that the main goal was a spatial analysis, not really considering the temporal aspect. I would say that as long as you have time-specific measurements of water quality, of natural, and of your diatom counts, that you should be able to apply this to look at temporal variation in MMI scores. There's a little bit of, if you go way far back, then the NHD landscape data might not be applicable. If you're looking at 100-year-old data, then current land, natural covariates might not be descriptive of those sites. But for most studies that are looking within the last few decades, that should be fine.
And just to follow up on mentioning the NHD data sets, Sarah points out that the Shiny application is currently based in North America only, and so I suppose the ecology only really applies to North American taxa. And correct me if I misspoke there. Luisa has a question. If you have undescribed taxa, how will they get treated after you've harmonized the data? Will they get dropped from the analysis?
Currently, yes. So that was in that step 2 process where I showed that Encyonema temperae, where it didn't have it, it's an accepted name, and we observed it in Colorado, but it didn't have a value for any of the metrics in the database that's used in the MMI or to construct the MMI. Therefore, any counts of Encyonema temperae were completely dropped from analysis, therefore limiting some of that information that you could have gained from that taxon. And the same is true for unidentified taxa like Ecmanthidium species 1. Currently, they're all dropped, and we don't have a way of adding those metrics. We don't have a direct way of saying, oh, well, we should add in metric values for this taxon 1 because we might not know enough about that taxon, and 2, we just need to open up those channels for users. But perhaps that's something we should potentially implement, is if one study is finding they have one taxon over and over and it's in high abundance and it's getting dropped from the analysis, then we should potentially revise or add that taxon into the metric table if possible. But that's conjecture at this point.
Great, thanks. And I think you strategically gave a very long talk, so we'll have a short Q&A session. And I have one more question for you while we wait to see if anybody else asks. And while I ask this question, I'm going to ask you to grab your guitar and play a little 10-second riff for us here at the end of the talk. But so what I wanted to ask you, and I saw some comments in the chat, is whether or not someone might be able to use the Shiny app just to harmonize their own nomenclature. I mean, maybe they're not interested in using the MMIs, but maybe they want to align their taxonomy with yours. I know there's some R code and different ways that you can do this. Could you do that with the Shiny app? Could you just check your names by throwing your data into the Shiny app?
Yes, you can. It's might not be the most accurate way of doing so, because what you're harmonizing your names to taxa that we have metric values for. And like I said, for the most part, those are the most updated names, the most taxonomically accepted names, but there might be some deviation from that. So I would still, so it could be a good pass, but you're also going to be doing some guesswork because we don't connect your name to potential matches. We just say that name's not in the database. It's up to you now to see what it could be. So like I said, this biodata in the chat, this biodata check function in R created by Ian Bishop, formerly of Sarah Spalding's lab, now at University of Rhode Island, is, that's a great tool of updating names as well, and might be a little bit more nuanced than using the Shiny app.
Thank you. I have another question for you, and this comes from Isan. If I'm saying that correctly, sorry. Please correct me if I'm not. He says, "Hi Nick, is there any similarity with PIBI, P-I-B-I? I'm unfamiliar with this tool."
Is that the phytoplankton index of biotic integrity? I know it by name, but I'm not sure the mechanics of how it was constructed. So. So if it's an MMI, then there would be some similarity, but there aren't a lot of MMIs out there that have this predictive modeling side of being able to account for natural variation. And again, if it's a phytoplankton-based index, paraphyton index, my apologies. So I'm not familiar with that. Maybe Sarah, do you have any insight into the paraphyton IBI and how it compares? I expect it's the same general concept, but that there would be differences in the construction, as I discussed early on in the introduction of the Carlisle et al., how it accounts for taxonomic inconsistency, it accounts for kind of the number of component metrics, those types of things, which previous IBIs don't have as much work done on there. Not saying that they're completely invalid, but that was one concerted effort by this Carlisle et al. MMI that might be different from things like the paraphyton IBI.
Isan, are you talking about the one from California? That index? If so, okay. Yeah, so that one also incorporates soft algae as well as diatoms. And it is a MMI, but it doesn't include the modeling for natural variability, so not a geographic context.
Leila asks, have you tried to use qualitative diatom data, or can you use qualitative?
Yeah, I wonder if you could expand on what sort of qualitative diatom data. Is it kind of general estimates of relative abundance of different taxa? Is it differences in the taxonomy, like the taxonomic lineage? I guess I'm unclear on what sort of data you would be able to input in there, because my understanding is that you really do need some estimate, at least, of a taxon and some value that's relative to the other values for each taxon. And it could be, it could be rough estimates of the number of taxa, but just know that that would likely limit the performance of the MMI itself as well. You could do it, but you might want to use caution in the interpretation. Oh.
I don't know if it is presence, absence, or not, but
sorry. Hi. Well, what I wanted to know if you tried to check just with the presence and absence of taxa, and so to and so to see if you can have the same response that you had with the phosphorus and conductivity. Thank you.
Yeah, thanks for the question. I think their presence absence, there are some metrics that are based purely on presence absence of taxa with values for a given metric, but the MMI is also built on things that require proportional abundance of taxa, in which case I think that the MMI, certain individual metrics could perform well. So you could download the metric sheet and look at those metrics that have PA after that, or sorry, I'm not sure off the top of my head, but there might be a way of doing some sort of backdoor interpretation, but the MMI as a whole would likely not work with raw presence absence data.
Thank you.
Yeah, if I remember correctly, there was this older software called ADAS. I think it was developed by the USGS, and there was the ability to use quantitative or presence absence data with that, but maybe it didn't apply to all of the metrics. So I guess I'm going to be a good host here and wrap things up. Thank you very much, Nick, for your wonderful talk today. One thing to just point out, Hera asks if your tutorials will be available, and I think the answer is yes. They'll be up on diatoms.org.
Yes, so the link I shared earlier of the project of the Carlisle MMI will upload all the videos that were in this presentation. You couldn't tell the difference probably between some of them, but the walkthroughs of the web interface will be uploaded separately, as well as the two that I mentioned but didn't go through, which was determining COM IDs and drainage area. Those will all be made available on that page, and I'll post that link here in just one moment.
And we'll also have a recording of your recording available.
Exactly.
All right, well, thank you very much, Nick. Wonderful talk and super cool app. I can't wait to work in some streams and use it myself. Yeah, thank you very much, Dr. Schulte. Everyone have a great day.
Great. Thank you so much, everybody.